Hello, everyone, and welcome to SOI's first Back Ashore episode of 2024. Back Ashore is a discussion series where we revisit with scientists who have sailed on Falkor and learn what has happened since they completed their at-sea expedition. My name is Allison Miller, and I am the Senior Manager of SOI's research portfolio. In 2020 and 2021, I can't believe that's already almost 4 years ago, RV Falkor circumnavigated the continent of Australia, conducting oceanographic research in canyons, reefs, and seas surrounding Australia. The research led to some unprecedented discoveries, first ever sightings, and comprehensive surveys, including the first study of northwestern Australia's deep corals, the first seafloor map of the UN ocean decade, discovering a 500-meter tall coral reef in the Great Barrier Reef, and discovering new corals and sighting of an estimated 150-foot Siphonophore, thought to be the longest animal ever recorded at the time. And that's just a list of what happened while these expeditions were taking place on board the vessel. And today we are joined by 3 scientists who participated in a number of the expeditions around Australia, and we can't wait to hear their research updates tonight. Dr. Nerida Wilson from CSIRO led an expedition to the Ningaloo Canyon in early 2020, and then also sailed as a scientist supporting research in Ashmore Reef in the last expedition in Australia in 2021. Dr. Rob Beaman, joining us from James Cook University, led our first ever virtual mapping and exploration expedition from his home during the pandemic, and also sailed aboard 2 expeditions in the fall of 2020. And Dr. Jeremy Horowitz, a Peter Buck postdoctoral fellow at Smithsonian's National Museum of Natural History in Washington, D.C., was a PhD student at James Cook University at the time he sailed on board Falkor in 2020. We're going to start by letting each scientist introduce themselves and the science that they were conducting on board research vessel Falkor, and then we'll jump into some questions. I have some prepared focused on the outcomes of these expeditions that I'm really excited to hear, but I'd like to remind the audience that they can put their questions in chat and we'll make sure we get to those as well. Nerida, I'd like to start with you. Would you like to give a update of the expeditions you were on board Falkor and introduce yourself in a bit more detail?
Thank you. Thanks for having me this morning. Excuse me, it is this morning, so I have a froggy throat. So in 2020, I led an expedition with a team from the Western Australian Museum and a whole lot of other institutions. I mean, most of these expeditions are supported by a huge number of different people on board and off board. And we went up to a place called the Gascoyne Marine Park. And in that park, there are 2 canyons, Clotes Canyon and Cape Range Canyon. And this area is adjacent to the Ningaloo World Heritage Area, which is really, really diverse. So there hadn't been at that time any deep sea exploration of those canyons. And we were, anything that we saw was going to be new and provide new understanding. So we did a biodiversity survey, excuse me, in that area and had a wonderful time. And did you want me to also talk about Ashmore at this point?
Yeah, please go ahead and talk about them both.
Sure. And so then the following year, I was then lucky enough to participate on a cruise led by Karen Miller at the Australian Institute of Marine Science. And we went and looked at the mesophotic communities around Ashmore Reef. And so these are the areas that aren't sort of not super deep sea, but in that sort of 50 to 200 meter zone, which has limited light penetration. And again, to that point, there had just been no way to explore or measure anything in that zone at all. So it was a really interesting time. A little is known about the shallow regions there, but of course, always the deeper you go, the less you know. So it was a really great opportunity to explore that area.
Thanks.
Yes.
I'm reminded of all the beautiful imagery that you saw on both of those expeditions, but I know that was just the start of the research outcomes before you left the vessel. We'll go over to Dr. Robin Beaman, who was on board a number of times and also led an expedition not on board. Rob, could you talk about some of the goals that you had both virtually and while you were on board the vessel?
Yeah, hi Alison, and to anybody watching. Yeah, it was 4 years ago, a very intense period. It was just the start of the pandemic. You mentioned that that first one was done remotely. It was quite a period of upheaval. We were all locked down at home, and then suddenly the Falcor turned up in Cairns, having had all of its pre-planned voyages cancelled post Nerida's Ningaloo Canyons voyage, and we worked very hard to keep the Falcor within Australian waters, because at that point there was vessels were being banned out of Australian waters. But it gave us an opportunity to spend, well, it turned out to be a month and a half doing what was called the visioning the Coral Sea Marine Park. And it came about because with all of that sea time that had been cancelled, I was asked by Schmidt Ocean Institute, could I put together an expedition with the proviso that it would all have to be remote? So we all swung into action. We brought all our colleagues on board, and every day we would basically plan as if we were on board what the day's work would be. It was primarily a mapping expedition to begin with, but as we got, I guess, more in the groove, we found that we were able to do more and more ROV dives. And so out on the Queensland Plateau, it's the world's largest carbonate plateau. It's part of the Coral Sea Marine Park, one of this great network of marine parks around Australia. But it is an area that hadn't really been explored in the deep, just echoing what Nerida just said. And so by mapping around the flanks of these 30 large atolls and then putting cameras down, it was a revelation to find not only that three-dimensional picture of the seafloor, but actually what is living down there. And so we found that in that mesophotic zone, this low light area, but in the Coral Sea, the water is extremely clear. We were getting corals down to, like zooxanthellate corals, down to about 114 meters, 140 meters depth. And then it would change into a different environment. So there would be large foraminifera about the size of a dinner plate just scattered across the seafloor. And so we actually had never seen that before. And then as we got further deeper down, it was all that lovely cold water environment. This is a tripod fish that is sitting there. Not as much life, of course, but still just really astounding to be there on shore and remotely watching live and speaking to all the ROV dives that were happening. It was just an amazing experience.
Rob, I'm so glad you were able to help us bring science to the public while we were all at home. It was a really great.
That then
Go ahead.
led to the series of all the others that came after that. So I was actually involved with 6 back-to-back voyages after that visioning the Coral Sea, and so we went straight into another expedition. This was the corals and canyons of the Great Barrier Reef and the Coral Sea. And this is one that was planned, but it almost didn't come about just because of all the restrictions on transport around the country. But it was a large voyage involving a lot of colleagues from Geoscience Australia, led by really close colleagues. And we managed to get quite a few scientists on board. Jeremy was there. Jeremy will have his chance in a moment. But we were actually then able to build upon the previous voyage out on the Queensland Plateau, and also put cameras down in the deep Great Barrier Reef, which really had not been done before. So Great Barrier Reef is world's largest extant coral reef system. It's a World Heritage Area. The shallow coral reef systems are actually really well understood and studied, but beyond about 100 meters depth, very little was known. So this was the first opportunity to put cameras down along the edge, the great big cliff that kind of aligns the Great Barrier Reef. And there's Jeremy in the background. But it was great to have all these marine geologists and biologists on board. And again, it was a fantastic experience. That led to the next one, which was one I was directly involved with the northern depths of the Great Barrier Reef. So some background: the Cape York region of the Great Barrier Reef is the very far northern part, extremely remote. Very few people go up there. Certainly, no one had really studied what was off the edge of the coral reef, the shallow coral reefs. There'd been no mapping done with any modern technology. And so it was really a voyage into the unknown. But I've been trying to get vessels up there for many years, and this was just a golden opportunity. So with a very skeleton science crew, because at that point, lots of restrictions on transport in Australia, we managed to do extensive mapping over about a month and a half. So we did a series of ROV dives. This is a ram's horn squid, the first time it actually been seen in situ in the water column. We wrote a paper about this with Dougal Lindsay from Jamstec, who was remote speaking through these incredible midwater dives that we were doing. But we were able to map the edge of the Great Barrier Reef, and I can easily say it was the most complex and spectacular seafloor I have seen anywhere in Australia. That far northern part of the Great Barrier Reef is truly spectacular, including finding this 500-meter tall detached coral reef, which no one knew in existence. So here's a 3D version of it. It's that spike right in the middle. It's like a great big spire. It's 500 meters tall, so it's taller than the Empire State. It's taller than the Eiffel Tower, taller than the Sydney Tower. 500 meters tall. It's a significant feature, but no one even knew it existed. So it's blade-like in view. And we were able to sail over the top and actually do an ROV dive on it. So that day was quite memorable. It was like climbing a mountain. And we now know exactly what's at the base, what's living as we go up into the mesophotic zone, and then actually what's at the surface. It's a living coral reef. It's very healthy and thriving, covered in fish and sharks. It's acting very much like a giant underwater seamount, just on the edge of the Great Barrier Reef.
I look forward to hearing more in our conversation, but I want to let Jeremy introduce himself. He was a student on board in the summer and fall of 2020. Jeremy, could you talk a little bit about what your goals were while you were on board the vessel working with Rob and other scientists?
Yeah, thanks, Alvin. So to go right off of what Rob was saying, and to basically echo what Nerida was saying, the Great Barrier Reef and Coral Sea, so little was known about what lives in this huge reef, this really big area. And I guess previous to our time on Falker, there was an expedition called the Sedaris Project. And Robin knows a lot about this expedition. We actually wrote a paper about black corals collected from this expedition. It was 30 years ago, and they trawled and they collected basically all of what we at that time, prior to Falker, knew about biodiversity in these habitats. And I guess there's pros and cons to trawling. We were able to figure out what was there, but trawling damages the corals that are collected, and it destroys a lot of the tissue that we need to extract and sequence DNA. So when Falker was, in our part of the world for so long, and Rob was able to very quickly put together these amazing expeditions, I was able to, just kind of fortuitously be on board and work on the biology that we collected. So we spent a lot of time, watching what was the ROV was seeing, finally seeing for the first time these deep sea corals, understanding the habitats that these different species live in. And we learned a lot just by seeing how these corals were attached to the sediment. Here's a really good example. This is a new species that was described a few years ago now, Hexapathys bykovskii. And I think it was Rob actually who first said that this is a relic species growing on a relic species because it was growing on a nautilus shell. So it was an amazing, this is probably the best moment that I, remember of my time on Falker, because, we went to collect this coral and we thought we were grabbing a rock. But I mean, because the ROV pilots were so amazing, they were able to, without even breaking the Nautilus shell. So it's at the Museum of Tropical Queensland now. It's it's a holotype for that species. And I mean, if it was trawled, there's no way we would have gotten in that condition. We wouldn't have been able to sequence it. We probably would not have been able to determine that it's a new to science species. So just having all of this time at sea with this, great group of scientists and the great crew, we were able to go from trying to understand biodiversity through like surrogates, in the past, before we knew what was living in these habitats, we would say, okay, we think this habitat is at this depth. We think these species occur at these depths. Therefore, we're going to estimate biodiversity to be this. But now we've got real, we collected specimens, we're identifying specimens. And so we know what lives in these habitats. And in the end of the day, all that means is we're able to make more informed conservation decisions. So, I mean, this is a once in a lifetime opportunity for Australia and for, the Great Barrier Reef and Coral Sea to understand and conserve biodiversity. So it's just lucky to be on board as a PhD student.
Thanks, Jeremy. You guys all have me already excited for all the science that came out of these expeditions, and we're only 19 minutes into our conversation and have barely touched on what you've been doing back at shore. So I'm excited for this conversation. Nerida, I wanted to circle back to you. And when you left the dock in March on Falkor, you essentially returned to a different world in April. And I was wondering if you could talk a little bit about how coming back to COVID and a lot of things shut down impacted your plans for getting samples into the lab and beginning the work on shore that I imagine you were very excited to start and had a bit of delay getting started on everything.
Yeah, I think it's an absolute understatement to say everything changed. And not just when we got back, but actually for our cruise plan as well, because we had, it was a 4-week expedition and we had planned to change out people about halfway through to allow different people with different expertise to come along and contribute. And unfortunately, that change out was no longer possible. And so quite a number of people on board had to make a decision about leave and try and get home, or they might be shut out for unknown periods. It was a very unknown time. So some people fled and no new people were allowed on, and the remaining people pulled hard to complete the expedition with all of the incredible sampling and work that needed to be done. And of course, at the same time we're calling home to friends and family and hearing these crazy stories of people lining up for food and, all of these difficulties in getting just the normal things for life, let alone science. And yes, as Rob had mentioned, transport shutting down and borders closing. And so there were, lots of, I feel like every day was a new plan. Like certainly, you as Rob was saying, you sort of plan the day's work, but we were also planning sort of escape paths for people and all kinds of interesting things. And every day there was some other bit of news that meant we'd replan everything. So it was let's say it was a very agile cruise. But of course, we were also just completely enthralled with the work that was going on. So it was also easy in a way to lose ourselves in this sort of science escapism where you just got to make these incredible discoveries and do all this amazing work and then try not to think about what's happening out there. But of course, every trip comes to an end, and unfortunately we had to come back. We could have stayed up forever.
Were you able to get into your lab and drop your samples off, or did you have samples living in your home refrigerator for a while?
No, fortunately, we were able to bed down the samples safely straight away. So that was very, very comforting. But some of the work, just couldn't happen for a long time. And I think, I think most people understand that taxonomy is quite a slow kind of work anyway. But one of the things that you really need to do is to get in a lab and access certain bits of equipment which might be living in, at a university or in a different institution, and all of those sort of cross visitation things, or trying to access holotypes, or even just remotely, like asking someone overseas, could you please take a photo of this thing? It just all was impossible. And there was another interesting sort of thing, that when when some of those restrictions did lift, I think, a lot of people made some different life choices afterwards. So everyone's work patterns really changed. So yeah, there were lots of challenges to getting some of that work done, and some of it still goes on because life goes on and people move and their situations change, and so yeah, but taxonomists are a patient bunch, so it goes on.
Nerida, can I just jump in? One of those pictures, it looks like it's an octopus with a scrubbing brush. It looks like you're about to clean its teeth. Its beak. What's going on there? Tell us.
Yeah, look, this was absolutely one of my most favorite things on the cruise, and it was just a little thought that I had before we went. Like, what if we see something that's too big to fit in the sampling equipment? Like, who knows what you're going to encounter? And I was thinking, we just need like a little swab. Little did I know at the time, right, how much we would be swabbing our noses and mouths for the next while. Anyway, I wasn't able to find something that was sturdy enough. I was going to sort of medical supplies and things like that. And then I think the night before, or the second night before we left, I was in the supermarket and I just thought, you know what? Maybe a scrubbing brush will work. So I just bought an armload. And so we had this idea that essentially, for some of these very mobile animals, we could simply kind of swab them to get some tissue or some mucus that would contain some tissue that we could do some sequencing and get some DNA from that animal. And fortunately, cephalopods are very inquisitive. And so if you present them with a brush, they will often grab it and poke it and touch it and kindly wipe their mucus on it. And so we had some very, very successful deployments of this brush, which the online community dubbed immediately the kitchen brush of science, KBOS. And so, which has been widely adopted. It's fabulous. But yeah, we were able, I think, out of the 5 deployments, we got sequence from all 5. So it was very successful, and just one of those fun kind of, combining a very low-tech approach with a very high-tech piece of equipment holding it. So yeah.
Yeah, I love that. Rob, you mentioned leading our first virtual expedition. What was the planning and execution like for that? And how were some of the planning activities similar to what you would be doing if you were on board? And what was different besides obviously not being on the vessel itself?
Yeah, it takes a real mental shift. I would 100% advocate it's better to be on board the ship. It's just a lot easier. You have the rest of the world away from you. It's being on board a large ocean research vessel, which I love doing, is you're fully involved with it. You just sleep, work, you do your shift, and it's great to interact with everyone on board to the exclusion of the world. But being on shore was, as Nerida pointed out, it was quite disruptive. That said, I absolutely loved going to sleep at night and knowing that the next day I would be virtually on board the ship. And so we would have our remote meetings. They were just like we would sit in the conference room talking about the day's plans. We have to have plan A, B, C, D, but we were doing it in real time. So the other thing was being at home. I've got a disabled daughter. She's quite hard work. I found working with the Falcor a really nice distraction. So there was that nice part of it. But remember, none of us had done it before, and that works both ways. Neither the Schmidt Ocean Institute or ourselves. So we kind of had to figure it out. We couldn't have done it without satellite, that broadband satellite on board, but I think it was very successful. What I really enjoyed was the fact that we could engage with all these other scientists and the general public from around the world, which never happens usually on a traditional voyage, where you go out with a big group of scientists. You might write a few blogs, you come back and you tell the world what you did. But in this case, we were interacting with everybody else who was also locked down at home. Suddenly, there was the whole world watching, or felt like the whole world watching, at the same time we were. So I was learning a lot. It was all of those coral experts that, I'm no coral expert, but to have them interact with us in real time and ask questions as we're going along, that was a revelation. And of course, it got better as we got more experience and went on. But so it was two sides. But I mentally had to shift from being at sea. I was still able to go down to the cafe and grab a coffee. That was a really nice thing. But my wife would often say, "So where are you, Rob? Are you at sea or are you at home?" And I'd go, "I'm at sea at the moment." And, and then I'd have to shift back to being at home. So, it was quite a mental juggle.
Yeah, I imagine that's funny. Although I think sleeping in your own bed might have been a positive of that experience as well. At least for me. Jeremy, sorry, go ahead. Yeah.
Just one last little bit there. We were watching the mapping in real time. Now, this had never been done on any expeditions I've been on, where I was actually able to see all the multi-beam systems in real time. And so we had that virtual ability for me to see where the ship was mapping in 3D. And so we could guide where the ship was going, or there was that constant discussion as to, there's something in front of us, or we have to go around it, or the weather's bad. And so I was actually able to watch that mapping in 3D. So that had never been done before, and I thoroughly enjoyed that.
That's amazing. Did you take some of the experience you had while you were at home and bring that on board with you once you were back on board the vessel? Were there some lessons learned there that you translated to being on the vessel?
Probably, I think more so to point out to Australian research organisations that you can actually conduct virtual science and share it real time with the world. And, the Falcon was the great experience of that. And so for voyages that I've been involved with since, we have tried really hard to say, can you replicate what the Falcon was doing? Can I be on shore if I'm not physically on board and watch your mapping in real time? And we've actually been able to achieve this. So it was a benchmark that's allowed us to take forward into other voyages.
Great to hear. Jeremy, you participated in some black coral sampling that ended up being groundbreaking and kind of set you on the trajectory for where you are now. Could you, how did the expedition that you were on in Australia help lead you to where you are today back in the US?
Yeah, I guess I'll start by saying, if you ask, 95% of the people who were doing their PhD during COVID, they would say it negatively impacted their projects. I had friends, Kala, people who were doing the PhD at JCU at the time, and they had to completely change their project, after working on things for 2 years because they couldn't go back to their study site. But for me, it defined my PhD. So, right off the bat, I had months at sea on this amazing research vessel. And it's, for most people, it takes years to try to get onto one of these expeditions. But, because of the situation, I was able to learn firsthand how to do all of these things, work on not just black corals, but all coral groups and sponges. And I was able to collaborate with, world experts, again, during my early PhD days. And I think this kind of set me on a really good path to be where I'm at now. I was able to discover and describe my first species. I guess describing new species is kind of a numbers game. The more specimens you have the more specimens you look through, the greater chance there is that you'll find something that's new to science. And because I was on board, I was the first person to look up. I was able to identify, oh, let's grab this and look at it. I was able to describe a whole lot of species, from this falcor. This one on the right is a new species. This one on the left is a very common species. You wouldn't know by looking at the two. They look very similar. I was able to publish a few papers on describing species. I also was able to use this falcor data to study the evolutionary history of black corals. So, 400 million years ago, how have these species evolved with depth and morphologically through time? Here's this image is Antipathes falcoreae, which I named after, of course, falcor. And it has these really cool morphological spines. And there's nothing, most black coral spines are very simple. Like on the right, these are examples of different families of black corals, triangular spines. But a lot of this falcor specimens had these really wild spines. And so it shows a kind of evolution in these morphological features. We still don't know the function of these spines, but that's future research. The data that was produced from the falcor expeditions really it changed what we understood about species relationships. So, all the world experts on black corals, all 5 of us, we now have a new understanding of how species are related. And that's because we're able to collect and sequence a whole bunch of specimens. And here's an example of a pretty updated black coral phylogeny. And a lot of these are falcor specimens. And we were able to determine that species generally have smaller ranges than we thought. We used to think, species were cosmopolitan. They occurred in all oceans, really wide-ranging depths. And that's because they look really similar. But when you sequence them, you see they fall out in different branches. And this is like a family tree. And so you'd find things that look really similar, but are on different branches. And that indicates that they don't have a common ancestor. They're not related. And so it's an example of convergent evolution where things look a certain way because that way has some sort of function that makes them persist. It's really advantageous for them to look a certain way. So they do in these different habitats. So now when I'm at the Smithsonian Institution, I'm sequencing a whole lot of specimens, a lot of holotypes. So holotypes are the specimens from which species were first described. So these are the best representations of these species. And I'm able to sequence these holotype specimens, and I'm comparing them to the Australian black corals that we collected years ago. And I'm able to say whether, well, one, are they the same species? And if they are, how big of a range expansion, does the species have? Or is this a new to science species? And most of the time, yes, if they're so geographically separate, they're probably going to be different species. So I imagine my current professional trajectory would be very different without my time on falcor. So I'm very grateful.
Thanks. I love your enthusiasm. You just made black coral taxonomy very exciting. So I appreciate that.
It is exciting.
I'm not saying it's not, but you made it extra exciting. So I love it. Nerida, just to circle back to you, one of the first kind of products or publications that you put out after your expedition was an illustrated guide to the fauna of the Ningaloo Canyons, highlighting all of the life that you saw there. Was there anything you saw in the Ningaloo Canyon that particularly surprised you, or anything that you've learned about the life there while you've been looking at the samples and analyzing your data back on shore that surprised you or you're excited to share? Oh, you're on mute.
Almost all of it, actually. Because we just had so little idea of what was, what animals were existing in that depth. Almost everything was kind of a surprise, and it sounds crazy, but I think putting together the illustrated guide, which I'm going to say sort of is the wrong order that people normally do. So, normally people will do an expedition, they'll come back, they'll spend a few years working up the taxa and publish a paper on that. But we sort of embraced, I think, the Falkor sharing mentality, if you like, and decided to sort of put out this guide showing, most of the organisms that we had found on that trip before they had been really worked up. So some of them are not, identified. They might just be identified to family or genus. But in the hopes that it's a way for experts in other areas to see the material and might be able to incorporate it into their studies as well. So it was a little unusual, but I think, it really gets to the heart of that open data and open sharing of findings. So it was a really really nice thing to do. I enjoyed myself a lot. And they're beautiful. They're almost all beautiful organisms, right?
Yeah, I was going to say they are beautiful, and you put together a great guide there, and I love that spirit of openly sharing everything before you fully described it. Also, in the spirit of openly sharing, you, as well as all of our other dives, were live streaming to YouTube when we came across a giant Siphonophore. And we have an audience question on whether you or anyone got the actual calculated length of the long Siphonophore.
I knew somebody would ask that. Oh gosh, that has actually turned a little into the bane of my life. So, I mean, it was such an amazing discovery, but of course, as you might remember, it was quite serendipitous. We had been working on the benthic part of the seafloor, and so we had sort of completed our day's work. And we're on our way up when this was spotted by one of the pilots. And so obviously, we stopped and took imagery and, tried to, let's say, circumnavigate the organism without disturbing it, because you have to remember too that it's moving in 3D space. And so, we had to not get too close. We didn't want the thrusters to kind of, disturb the organism. So at the same time, I guess we were running against losing the light and being able to safely retrieve all of the equipment and get it stowed on board. So we had a pretty limited time. So the imagery that we have from that organism isn't the perfect data set to be able to fully measure that length. We've actually had 2 groups try to achieve that task of being able to kind of, I guess, recreate it in a way that you can, really robustly measure that. And we're not there yet, is all I'm going to say. So it has been, a frustrating thing. And I think because everyone is so interested in exactly how long it is, it's not a thing we can just, measure poorly. It's got to be done really, really well. So yes, still at that. Thanks for
good
asking.
luck. I hope the 2 groups working on it at least come to a similar number, so you know that they're on the right track. But thank you for sharing that research challenge that you're facing. Rob, you showed some of this earlier, but you produced a lot of fly-through videos and story maps of the new bathymetry that was collected of the Australian seabed. Could you talk about how the Australian government has made use of this seafloor data and how you shared it with the Geoscience Australia and other agencies in Australia?
Yes, certainly. To put it into perspective, after all of those voyages in the northeast part of Australia, it came to around somewhere around 200,000 square kilometres of new seafloor being mapped. So in Australia, it's just a bit under the size of the state of Victoria, a fairly large state here. And so it's an astounding amount of new seafloor data. One of the challenges of then is how you actually show it. And so very quickly within the Oz Seabed group at Geoscience Australia, they were able to take that data and within a very few short weeks turn it around. Now, it's not perfect, but they were able to turn it around and have it available for download on the AusSed Marine Data Portal, which is our main database in Australia, and it's a public portal. So those 3D data sets are downloadable by the public even now. That said, we are still going down this long path. Science continues long after the voyages of doing very detailed cleaning of noise within the multi-beam data. Remember, this is collected by sonar, so there's always going to be noise in it. So there's a lot of work going on to clean it. But when you're talking 200,000 odd square kilometres, there's still a lot to go. That said, we are kind of cherry picking parts of it to do more detailed science on. So, for instance, this that 500 metre tall drowned reef, that new one that we found. So we're trying to do a much better detailed 3D model together with the marine life from bottom, from 500 metres down all the way to the surface. A full detailed variation in the biology within that depth range. Ultimately, though, we want to actually spread that right across all of the Coral Sea and look at how those depth changes influence the habitat and the different species there. So there's quite a lot of work that goes on, and Nerida talked about it, this ongoing effort that happens long after the voyage is finished. So a good example is the fish work. So there was this, I call them the fish army. Some were on board, but most were ashore. And boy, have they done some fantastic work. So we're just about to publish about to put out for a formal review, a full comprehensive picture of all the new fish, the new ranges, the new species of fish life that have been discovered in the Coral Sea Marine Park. So that's happening. We're writing this up as we speak, but it's taken some years to bring in all these different experts in from around the world, some directly involved with the voyage, but others not, to really bring that picture together. So, the work continues.
Great to hear, Rob, and I can't wait to read that paper. So please make sure that you share it with us. Jeremy, you have had the exciting privilege of coming on board Schmidt Ocean Institute's new research vessel, Falkor 2, to study black corals in the waters of Puerto Rico. What did you learn in Australia that helped you plan or prepare for the expedition when you were studying black corals in North America?
Yeah, Falkor 2 is a really cool ship. Everything that's good about the first Falkor ship, I think they got input from all the scientists and just made it 10 times better. I guess my time in Australia on Falkor taught me how to be a really good field scientist. I learned a lot, because I had only been to sea I think once or twice before. But this was the first ROV expedition. And so I was able to learn from a lot of more experienced people. Actually, Rob taught me how to identify a glass coral. And he's a geologist. And so I learned by tasting it. So just so you know. So I learned how to image and preserve specimens while in Australia. And so when I was in Puerto Rico on Falkor 2, I brought that skill set with me. So we were able to take really good images on the ship. Something that often isn't done on expeditions, and it's really invaluable because the colors of these corals and the way they branch, they'll often change after they've been preserved. So when you get it on board, it's really important to do certain things to make sure you extract as much information out of these specimens as possible. I also became really close to the crew on Falkor because a lot of the crew were on Falkor and on Falkor 2. So those relationships led to better science. On day one in Puerto Rico, there was a student working on how much metal are in different types of corals. And I don't think we didn't have a method of making sure no metal touched the corals before they did their test to measure how much metal was in the corals. So I spoke to one of the pilots, J-Rod, on day one, and I said, look, this is something that we're trying to do. Any thoughts on how to do it? He goes, leave it to me. On day two, he had 3D printed this plastic wedge that was really sharp at the point, and we were able to hammer it down to break the coral to ensure that no metal had touched the coral. So I thought that was really amazing. And anytime you need something, the crew is always really helpful. So me knowing them before the Puerto Rico trip made it really easy and made me comfortable to ask for assistance. And I guess I also learned how to mentally prepare for time at sea. It could be challenging. You're in a fairly small space, although by no means are these ships small, but you're still with a group of people. And, I guess I kind of developed this positive attitude mantra, prior to these expeditions, and it leads to greater productivity and wellness. And, it's really important when you're, with a lot of people that everyone's really happy. So I learned my time in Australia how to come at it with a positive mentality, and it really makes you enjoy your time at sea and makes you enjoy collaborating and working really hard to get the jobs done. And I guess lastly, my time on Falcor in Australia, I got to know the citizen science community, and they watch as they consume as much ROV dives as possible. So these people know often more than the scientists who go on the ship, because they listen to every expedition, and every expedition has these specific aims and these real niche experts working on these different groups. And so the people who watch these dives, they learn all of this stuff. So when we're on the ship, we can get input from this citizen science community, and they're saying, when they were in Western Australia, there was a nudibranch that was identified as this, so invaluable resource to have, the people on shore, non-scientists assisting, because they bring to the table a lot that scientists can't. It's really great including them in the Puerto Rico expedition as well.
Thanks, Jeremy. Thanks for that shout out to our amazing crew. None of this science would be possible without them. So we're very grateful for all the hard workers on board. And I have to ask, what does a glass coral taste like?
Do you want me to answer it, or should Rob answer it? It's glass.
Anyone who knows what it tastes like.
It's glass. So, if you've ever bitten into a piece of glass, I'm sure we all have at one point in our lives. It's exactly what it tastes like.
It's crunchy.
All right, good to know. Nerida, we have a question for you from our audience. I suspect this may have come from one of the super fans that Jeremy was talking about. So thank you for your question. But do you have any updates on the Megalocrantia that was sampled with the KBOS on dive 337?
I love a good specific question. Yeah, so I mean, I can't speak particularly of that specimen, but we certainly are in the process of preparing a publication that goes through all of the cephalopods that were found on that trip. And so that's being worked up in conjunction with Dr. Amanda Reed, formerly of the Australian Museum and now also at CSIRO. And so, yeah, so we're working on bringing together not just the KBOS results, but all of the results of the cephalopods that we found. So I'm really excited about that work.
Very exciting. And I also wanted to give you a chance, Nerida, to talk about the other expedition that you were on with Dr. Karen Miller to one of Australia's most remote marine parks, the Ashmore Reef. Do you have any outcomes of this expedition to share?
Yeah, I guess, part of the goals of that expedition were to not just to sort of look at the mesophotic region, but to kind of create some baseline information and to explore different ways of monitoring and measuring different parts of that ecosystem. And so there was an honours student at the time on board, Amy Kamiani, and she has published her work on the ecophysiology of some of those hard corals. And so she was able to look at the diet of different species and kind of figure out which ones are still relying on symbionts and which ones are using more external food sources. So yeah, so that's work is out. It was a really impressive piece of work, actually.
Was. Well, we're just about out of time. And before I give our scientists the last word on what they're working on now and what they're excited about, we did have a question about when is Schmidt Ocean Institute returning to Australia. So I have a map behind me. I'm trying to move out of the way, but I did want to let everyone know that RV Falkor 2 is planning to return to Australia in 2032 and 2033. So it sounds far away, but I think we can all agree it will be here before we know it. So get your scientific questions and ideas ready, and we'll be taking proposals for those in no time at all. So we're really excited to return and keep studying in the waters of Australia. So we have about 5 minutes left, and I just wanted to open the floor to any of our scientists before I wrap up and thank them. If you'd like to share what you're still working on with the data, anything exciting or just anything that you would like to share. And Rob, you and I were chatting a little bit before we went live about this, so I'll start with you.
Yeah, I'm working on Falkor data every day, Alison. There's mountains of it. And the thing that I'm particularly working on at the moment is generating high-resolution 3D models for the Queensland Plateau around all of these 30 large reefs. And since the Falkor, there have been another voyage. It's run by the German RV Sonne. And so I guess where the Falkor data has led us is from initially a voyage of discovery, like what is even there. It's now spawning a whole lot of other expeditions that are far more targeted. So the Sonne came in and they did a big seismic survey, basically edge mapping where the Falkor had been. So by giving them the data, it allowed them to better target where their expedition went, which was in 2022. We have a large expedition planned next year in 2025, run for Parks Australia, which is the body that looks after the Coral Sea Marine Park, and CSIRO. It's a biodiversity and fish study led by some of the people who are involved, Will White and John Pognosky, Fish Army from the original Falkor voyages. So these are more targeted. And the Australian Hydrographic Office has a real interest now in the Queensland Plateau to discover some of these drowned reefs that we found during those Falkor expeditions. So there's another big Commonwealth-funded expedition happening within the next year out on the Queensland Plateau. So it's spawned all of these future voyages. More data for me to play with, but I am still loving playing with the Falkor data.
Excellent. Sounds like every day is a new day and a new puzzle to solve with the data. So I love that. Jeremy, would you like to share anything that you're working on?
Sure. We are in the middle of conducting a formal taxonomic review of all black coral of the whole order. So there's currently 7 families of black corals. There will be 8 families in like a month. There'll be an 8th family in like a month. From Falkor 2, actually, we collected a new species that ended up being an entirely new family. And there's probably, based on the data, a lot of this comes from this tree. The species names were removed from this tree. But there's probably going to be another 7 families described. And most of the Falkor specimens will fall into these new families. So it's a lot of work. It's a collaboration with all the other world's black coral experts. Most of the experts, they work on specific families or from specific parts of the world, but together we're working on this really big review. And in the next maybe 2 or 3 years, if I focus really hard and if I can keep the current job that I have now, I'm trying to stick around at the Smithsonian Institution as long as I can. We'll be able to do it. And so, yeah, that's where I'm at right now.
That's very exciting. I'm looking forward to a new paper where we can welcome a new coral to the family. That's really exciting. Nerida, would you like to share some of the outcomes that you're working on?
Yeah, so for those who know me well, they'll know that at heart I'm a slug person. So I was absolutely thrilled with the amazing diversity of slugs that we found in the mesophotic zone at Ashmore Reef. And so I have a lot less time now than I did, but yeah, so essentially trying to identify those organisms. Many of them look externally quite similar, but for sequencing, you really need to look at shallow water representatives and, if available, other mesophotic representatives from around the world. So gathering all of those specimens and sequencing them is taking a little while, but that's the work that I'm super excited about. So yeah, it was way more slugs than I ever thought we would see. So super pumped.
That's very exciting. Well, thank you all so very much for taking the time to share your outcomes from these expeditions, from Falcor's time in Australia. I'm really excited to hear about all of the great scientific work that will keep coming out of this research. So please do keep us at SOI updated on all of the outcomes so that we can share them with our audience that's on here today, and also with the general public and our super fans. So thank you once again, and I look forward to hearing all about your scientific research outcomes in the near future. Thanks everyone online as well.
Thanks so
Bye.
much for giving the
All right.
opportunity.
Yeah, thank you. And thank you to the super